noisy thermal environment. And it can do this in a very, very precise way to really control outcomes that are very, very distinct and in some cases irreversible. So I think a very interesting question is really how can robust and yet programmable behavior at the cell scale arise from these noisy angstrom scale signals? So, there's been a lot of work done on this problem because it is signaling is an important problem, right? And broadly speaking, the question of allostery, I think for the purposes of this talk, we can just think of it interchangeably as the property of communicating signals through the medium of a protein. And the first thing to note is that there's been a lot of work, much of it by Rama's group, of really understanding how this coupling and this information encoding is already encoded in the sequence coevolution, right? So this is taken from the paper in 2011, really showing that you can actually get pretty accurate allosteric maps from looking at patterns of evolutionary coevolution between pairs of residues. And from that, you can construct these sectors that are these contiguous regions of the proteins that are co-evolving. And for the purposes of allostery, these sectors, in many cases, seem to be pretty predictive of what is actually what parts of the protein, in this case experimentally here labeled in red, can actually couple to the functionality of the protein. And you can see that for the most part, these allosterically active parts of the protein are connected through these co-evolving networks. So the information is encoded. Yeah, go ahead. Yeah.
Can you just tell us again what the blues and the reds are supposed to represent?
Yes, yes. So quickly. So the blue here is this sector, which is this set of residues in the protein that are co-evolving, right? So, and this sector is computed, right, from the statistics of looking at statistics between all pairs of residues in a protein in a sequence alignment, right? So that's construct. So this blue region is constructed from this alignment data or information about the sequences of proteins, and then the red are these hotspots in which, in their experiment, they're able to experimentally link this protein with another protein that is light activated and can be kind of tagged onto different parts of the protein to test if that part of the protein can allosterically affect the function. And then the red is just showing which parts are allosterically connected to the function of the protein. And this is just to illustrate that the sequence coevolution is a pretty good predictor of which parts of the protein are allosterically connected to the functional site. But I want to say that even though the information is encoded in the coevolutionary history, what I want to focus on is really looking at the question of, well, we know in physiological time this information is encoded somehow, right? In physiological time, the information is encoded in the structure and the fluctuations of the protein itself. And so what I want to focus on is looking at the protein as a physical medium that has to physically communicate this information in real time. How should we look at this problem of information communication? So the first thing that structural biologists will want to look at is obviously the static structure, the structure that you can look up on the protein data bank, usually a crystal structure of a protein. But for a lot of signaling proteins, in particular, the function of the protein is often not at all evident from the structure, right? And that is why things like coevolutionary analysis have provided such important information, because the function in many cases is not evident from the structure. So here I'm showing all of the human steroid receptors, right? There's different classes. And if you look at their crystal structures, right, and they're shown bound to their ligands, right, that they're supposed to detect. These all detect a family of ligands but they detect ligands that are different from each other, and they also have allosteric networks. That is, they have signaling networks and communication responses and networks that are different from each other. So they function differently. They are diversified in this sense, and they yet when we look at their structures, how they're folded looks the same, right? So you can reconstruct the evolutionary history of this protein family, right, by looking at comparing their sequence identity, and this is the construction of from their sequences, how this lineage is, uh, was created and diversified from previous common ancestors. And so the takeaway here is that if you were to analyze this protein family from the structure, so the TM score is um, a measure of differences in structures between pairs of proteins. If you were to just look at analyze the structural differences, and you try to like construct a phylogeny based on their physical structure, first of all, you would see that the signal doesn't have much dynamic range because these proteins are all basically folded into the same thing. But if you still try to use that to reconstruct the phylogeny, you would not get the correct phylogeny. So here you would predict that the AR and the MR belong together, whereas actually they're supposed to uh, uh, be separated one step prior, right? So the static structure is not then a very good even though the static structure is physically there as a physical representation of the protein, it is somehow not sufficient to then read out uh, the the um, the functional structure, if you will, of the protein. So, um, if it's not in the static structure, and obviously these proteins are transmitting information physically somehow, then of course the answer must be that it's in the ensemble of structures. It's in the fluctuations that must encode and transmit information for protein computation. It's the fluctuations that must encode the functional identity of the protein, and if you look at the correlations among the in the fluctuations, you might be able to predict the couplings that are inside the protein. But now, based on real physiological couplings, rather than um, uh, 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 co-evolutionary couplings that are, of course, constrained by physiology. Right?
Can I ask a question, Milo?
Yeah, go ahead.
Yeah, so just about this previous slide, so I understand that if you were just trying to look at the structures and using this matrix on the right, you would infer the wrong phylogeny. But then, I mean, if I were being naive, I would say, well, maybe I shouldn't have predicted the right phylogeny in the first place because, well, these different proteins that are even closely related could be doing different functions. Proteins that are farther apart could be doing more similar functions than proteins that are closer together. So
Yes.
like my naive guess would have been that I should say something about how close the ligands are that they would bind, as opposed to something about their sequence
Yes.
similarity, right? So is
Yes.
that a bad way of thinking about it?
No, that's perfect actually. And in fact, I put this thing here because actually as you say, uh, there is no default expectation that that the anything about the structure or maybe even the fluctuations should necessarily follow the evolutionary history, right? Like you might have mutations that are closer in sequence space and therefore are closer in sequence space. They're the next steps in the actual evolutionary history of this family, and yet they just happen to lead to larger perturbations in structure or dynamics, right? So in fact, this doesn't have to be at all true, right? And in fact, what I just said is not even if the structure, right, is not a good encoding of the evolutionary history, does not mean that the structure can't encode function actually, right? So that is a great question. That is not what I meant to imply. What I'm trying to say here is actually just planting a seed for something I'll show later on, right? because actually, in the field of structural biology, I think the viewpoint, right, of looking at the relationship between sequence and structure and sequence and fluctuations is such that the expectation is some kind of nonlinear, non-monotonic mapping that we wouldn't necessarily expect the anything about like the physical analysis of the protein to necessarily reconstruct the phylogenetic history. And I think that I think a main takeaway of what my talk is that we are sleeping on the information that's encoded in the fluctuations, and that actually the fluctuations actually encode a structure that really is capturing a lot of information that's missing. And one type of information is evolutionary information. So hold that thought for I guess a few slides down the line. But thank you for that. That's a great point. Just to reiterate, what the question is getting at, and what I completely agree with, is that just because structure in this case does not reconstitute phylogeny, does not mean that structure is not sufficient by itself to encode function, to encode function. Okay. The fact, though, is that structure by itself is not just empirically not sufficient to predict function. For example, the structure cannot be used to in any way to directly infer the allosteric sensitivity for a protein, right? So that is a fact, but that's a fact that's not like demonstrated by this slide. So that's a very important clarification question. Does that make sense?
Yeah, thanks.
Milo, just one more
Yes.
question.
Yeah.
I'm not quite sure how you're creating a phylogeny of proteins. I'm more familiar with phylogenies being created of like organisms. So could you like unpack that a little?
Yeah. So I mean, basically, you can construct a phylogeny as long as you have any kind of distance score between. So if you have a set of things, as long as you have a distance score between any pair of things, you can construct your estimate of like, well, just based on which things are closer and which things are further away.
The
Right.
distance.
So here you can have you have two proteins that have different sequences, right? So you can construct how far sequences are away from each other, right? And from that you can infer like the of the phylogenetic tree. The same with the if you have a score based on how different two proteins are structurally, you can use this distance score to also construct like your best guess of what is the phylogeny of these objects based on that distance score.
All right, thanks.
Great. All right. So okay, so empirically, structure in many cases is not enough to encode the function of what the protein does, and it does not capture what the networks of communication are in a protein. And the question is, okay, well, what if we look at the dynamics of a protein? If we look at the fluctuations, what can we do with the fluctuations? But once we get into the question of trying to capture some kind of measure of protein function or protein allosteric or protein intra-protein communication network using now the information from fluctuations, I think the question becomes, what are the right variables then to look at ensemble of proteins? And I guess in keeping in very broadly the theme of this meeting, it's really because the proteins are high dimensional objects, right? So how are we going to go about it? So the data itself here that I'm going to be working with, and it's the data that currently is really the only kind of data that has the sufficient both temporal and spatial resolution to do the kind of analysis that I'm going to talk about. But that may not be true for very much longer, right? There's experimental methods that can really get dense sampling of ensembles. But for now, the data source for generating these ensembles are molecular dynamic simulations in which you just simulate Newtonian dynamics of your protein surrounded by explicit solvent molecules, and you have a force field that parameterizes all the interactions in the protein for having to do with stretching of bonds, bending of bond angles, turning of dihedral angles, and of course these non-bonded Leonard-Jones and Coulomb interactions. This level of approximation is sufficient. This Newtonian dynamics is computationally very expensive, but is sufficient and to fold proteins, single domain proteins from scratch. So they have sufficient this method, or the force fields, are sufficiently well parameterized that this is a pretty accurate framework for generating ensembles of protein fluctuations. So here, this is a pretty old study now, just doing a survey of how well these molecular dynamics performs in terms of folding proteins from scratch. So the red is the crystal structure, I believe, and the blue is what the molecular dynamics simulations produce. But fine, let's take that we have an accurate data source for generating protein ensembles. But the question still remains, how are we going to extract the meaningful variables for predicting protein, intraprotein communication and protein function? Right. So one of the things to note is that if you were to look at, if you were to download a protein structure from the protein database, right, the way that protein structure is posited is actually in Cartesian coordinates. Okay. So, and in Cartesian coordinates, the kind of fluctuations and the correlations between degrees of freedom, right, they look very unimoto, many cases Gaussian-like. And that's really masking actually the underlying degrees of freedom that actually move proteins. Okay, so the degrees of freedom that actually move proteins are essentially these dihedral angles, these single bonds about which the molecule can rotate and therefore get a different configuration or different conformation. And so usually for a particular one residue of the protein, right, the two free dihedral angles in the backbone are called phi and psi. And then all the dihedral angles that have to do with the side chain of the amino acid, which varies depending on what amino acid it is, are just labeled chi1, chi2, etc. And these all can rotate. And these degrees of freedom are actually what gives essentially the structure of a protein, right? But because so what happens is that if you look at the, it's very well known for a very long time, right? It's if you look at the statistics of phi and psi, just the backbone, for example, you see that they occupy particular populated basins. This is called the Ramachandran diagram for looking at these dihedral angles. So that's been well appreciated. And if you actually just run molecular dynamics simulations over and look at what is the value of a dihedral angle over time for backbone as well as side chains. These are just two random residues of a protein in some molecular dynamics simulation. You'll see that what happens is that these dihedrals tend to stay in one base in one well-defined state and then make these discrete jumps between states, typically. Okay, and these jumps can be very frequent, or these jumps can have long persistence times. And so the underlying probability distribution of the residencies of dihedral angles is actually non-unimodal. It's a multimodal, very non-Gaussian. But if you were to just look at the Cartesian coordinates, the fluctuation look Gaussian just because of a incomplete version of the central limit theorem, right? So you have the way that Cartesian angles move is because of a superposition of many dihedral angles moving. of course, these dihedral angles are not moving independently. They're not drawn from an identically distributed distribution, and there's not like a infinite limit of them, right? So the central limit theorem does not formally apply, but the same principle that it tends towards is still at play here. And so if you look at the fluctuations of Cartesian coordinates, the fluctuations look unimodal, often very Gaussian, and so you can often characterize them just by covariances between degrees of freedom. But the actual underlying degrees of freedom, the dihedral angles, are multimodal. They look like this, right? They exist in different wells, essentially. And so if you look at a history, if you look at now, you can actually make a barcode movie of a protein, right? So here, the y-axis is time, and the x-axis are different dihedral, in this case, backbone dihedrals. Sorry, the dihedrals of the protein. This is a particular protein. I believe it was RAS that what we simulated, but doesn't really matter. We will see that if you now just plot this over time, you can now bin for each dihedral, you can bin them into specific discrete states, right? So here, if you choose 4 different states, you can call them the white, blue, green, and red states. And now you just ask which state this dihedral is in as a function of time. You then have these discrete barcodes for dihedrals. And this is a very good representation of the conformational state of the protein, right? You're missing all of the fluctuations that are happening within these basins. And so if you're ending up calculating entropy, you're going to be missing the vibrational entropy. But it turns out that the vibrational entropy actually just subtracts out. And what distinguishes different like states of a protein, right, is actually the configurational entropy, actually how many different discrete states are there, right? So this is then to first order a very, very good approximation of the state of the protein. But okay, that is only looking at individual dihedrals though, right? A protein residue has many dihedral degrees of freedom, right? And within a residue, they tend to be very, very strongly coupled, correlated. And then between residues, there's still going to be coupling, obviously, as well. So you can't just treat the individual protein dihedrals as independent, and you cannot treat different residues as independent. So then, how do we get a good representation? So you have this then potentially high-dimensional data. It's not just naively factorizable. We should probably look at degrees of freedom in terms of the dihedrals rather than the Cartesian coordinates. But how can we then map entire residues, right? So that we're talking about capturing the correlations that are very strong within residues, and then how can we reveal the correlated dynamics between residues? Because that's the kind of dynamics those are the kind of correlations that we would then use to predict like physiological coupling, direct coupling between different parts of the protein, right? To, for example, predict allosteric maps. So here I'm going to be talking about then some recent work from Caitlin, who's graduating soon in the lab, of a framework for doing this that leans on all of the observations I just talked about, but puts them together in a way that actually produces a self-consistent prediction both of the entire conformational entropy of a protein that we can test experimentally, as well as provide shared entropy maps within proteins, so we can actually then predict more site-specific aspects of a protein, like, for example, the allosteric sensitivity of different residues. So how does that work? So you first start out with an ensemble that, in our case, is generated from molecular dynamics simulations. And then here, the idea is that because within a residue, the dihedral angles are so strongly coupled, and by the way, it's not just dihedral angles, the bond angles are also able to bend a little bit too, and they turn out to be important. So, but for just simplicity, I'm going to ignore the bond angles, even though the bond angles are also part of all of this analysis, right? So let's imagine now, so what you do is that in the multidimensional space of all of the dihedral angles inside one residue, you sample the population density. And then you fit the population density using what's called a von Mises mixture model, which is basically just like a Gaussian basis set, but one that's on used for periodic boundary conditions, right? Because dihedral angles are periodic. So you use these components to optimally cover whatever probability distribution you have in the multidimensional space of whatever your dihedral is. If your dihedral is arginine, it has many, sorry, what your residue is, my mistake. If your residue is arginine, then it has many dihedrals, and it's a higher dimensional space, right? If your residue is glycine, it's a lower dimensional space, but you cover that space. And here you do it based on that particular residue in the simulation. So arginine in the core of the protein is going to be different than an arginine in the surface of the protein. After you your mixture model then covers or approximates that population density, then you cluster not in the original, not in the directly in the dihedral space, but you cluster in the components. So for example, these three components are in one cluster, and this component is in another cluster. And then all of these components that are like really poorly sampled, we actually just say that they belong to one state that's called a disorder state. Okay, so for example, in this particular toy example, that's just one dimensional. This would end up being state 1. This would end up being state 2, and then all this stuff would belong to the disorder state. Okay, so you would do that then. So for each residue in the protein, you would then identify these discrete states, not of individual dihedral angles, but of entire residues. And now you can calculate joint probability distributions between two residues. So if this is residue 1 of a protein and this is residue 2 of a protein, let's say residue 1 has 5 states and residue 2 has 4 states, you can get the joint probability distribution in this discrete states. So how many discrete states are there? So this is over multiple proteins that we looked at, but for most residues there are fewer than so the most likely bin is that there's fewer than 5 discrete states that for example, many residues are in the core of a protein and some of them don't really move at all, right? But there are some residues that have many tens of states, and these states need to be properly counted. These are these non-overlapping states, and it turns out in this high-dimensional space, they really are still non-overlapping, just like they're well separated when you look at the single dihedral. Okay, now that we have this, we can then calculate, for example, the mutual information between any two residues, right, based on the joint probability distribution. But if we did that, we would not be able to make a network that we can visualize, because if there's if there's n residues, there'll be an order of n squared pairs of couplings, and we wouldn't be able to visualize that. And furthermore, a lot of the couplings, just like the story was when people were analyzing couplings in sequence space, a lot of the couplings are not direct couplings, right? So maybe this residue and this residue are coupled because they are coupled through some intermediate. So the question is, okay, what if we actually projected this onto a spanning tree model, okay? And the reason for using a spanning tree is twofold. The first is that it provides us one way to estimate a sparse network, which is this tree network. But the second is that a spanning tree provides a straightforward way to calculate entropies of any subsets without any inconsistencies, right? So if it's going to be a spanning tree, there's this additional advantage that Cha and Liu worked out, that if you're going to use a spanning tree to, so let's say you have any arbitrary probability distribution over degrees of freedom, and you want to use some kind of a spanning tree um, approximation for that distribution, which means that, the way it's constructed is that, you have the probability of first degree of freedom 1, and then the probability of degree of freedom 1 and 2 is just the p of x1 times p of x2 given x1. Okay, so it's this times this conditional on this, and then the probability of the joint between 1, 2, and 3 is just you would just multiply p of x3 given x2, right? For example, if this is state 3. So that is the constrained model that a tree, the spanning tree, imposes. But if you were to use a spanning tree model, it turns out that there is always a best one, right? There's always a maximum likelihood one, which is equivalent to the one in which, regardless of what the original probability distribution actually is, which may actually not be well approximated by a tree even, right? The KL divergence between the probability distribution, the real probability distribution, and your tree probability distribution, right, is equal to the sum of the mutual information along the edges of your tree, right, and the negative of that, right, plus other terms that are independent of a tree model, of what, i.e., independent of which tree you happen to choose. Now, this thing could be huge, which means that, like, no tree is a good model for your data. Okay, your data is not well approximated by a tree at all. But even then, you can still choose what the best tree is, and the best tree is the one that maximizes the mutual information. So it's the maximum spanning tree of mutual information. Okay. Okay. So this is then the actual information structure, the object that we're going to use to construct the entropy maps of trees. So from this tree structure, we can directly estimate what the conformational entropy is, and that is the individual entropies of each residue minus just the edges, the mutual information on the edges of the tree. Okay. And so that provides a self-consistent way to calculate what the entropy is. And another thing it provides is that it can also give you the entropy map, because by just plotting what the entropy of residues are and what the shared entropy is between pairs of residues, that gives you the information structure of the protein, and you can use that to make predictions about, for example, allosteric sensitivity. So first of all, how good is a tree model? So what we can do is we can take we can calculate the mutual information between all pairs, and then we can calculate this maximum information spanning tree, and we can now ask for all the pairs of residues that are not edges in the tree, what is the ground truth mutual information that we calculated, and what is the mutual information that we would infer by propagating along the tree. If the tree was a really good model, we should be able to reconstitute the mutual information between any two pairs of residues just by propagating along the tree. And on a set of proteins that we looked at, the tree turned out to be a pretty good model for information sharing within proteins. Okay, so let's use this tree now. I'll go beyond self-consistency. Let's use the tree now to like predict experimental data. So one thing that we would like to do is to predict, just like the sequence coevolution methods have done, but now using the direct physical fluctuations of the protein, the allosteric sensitivity of a protein. So here's a kind of a workhorse protein that many people have looked at and have done experiments on, PDZ3. And here is the chemical physical structure of the protein. And here is the tree structure. So the size of the sphere is proportional to the entropy of a residue, and the volume of a edge is proportional to the mutual information or the mutual entropy between two residues. So this network then is a visualization of the information network predicted by this tree. And we can see, like, just like, qualitatively, it seems to find these hubs of allosteric sites, right? But actually, quantitatively, we can compare this to experimental data by looking at saturation mutagenesis data on this protein, and and basically asking, well, is the so for each site, so for each point is a residue, and each point has a um, uh, allosteric sensitivity, which is defined by if you were to mutate that residue into any of the 19 other residues, how many of those residues would have a larger effect on the uh, um, uh, activity, the function of the protein, than a mutation at the active site itself. Okay,
Can I
so
ask a question?
yeah, go ahead.
First, just to clarify, here you aren't using any contact map information for the protein, right?
No. So this is,
Okay, great.
I mean, this is generated based on, you
Right.
know, molecular dynamic simulations of the protein just fluctuating at equilibrium, right?
Cool. Okay.
Um,
And then
and
the second
yeah,
one, if I can ask,
yeah.
just so does this inference depend on how long you simulate for, or how long? Yeah. Just
Yes, yeah. So we found that in order to get convergence on like quantitative convergence for the these small single domain proteins we looked at, we need at least like 4 or 5 microseconds. But then like after that, like we get the same predictions like what the trees look like.
Thanks.
Yeah. Okay. So um, all right. So here so that is the experimental measure of how allosterically sensitive each residue is. And then the x-axis is the prediction from the tree, which we basically just say, okay, we ask what is the information shared between this residue and the active site calculated from the tree, and then we you know have we have a proportionality constant that we fit. Okay, um, so um, then quantitatively, um, the tree, not just kind of by the eye test that we look at here on the left, but quantitatively, we get uh, uh, good prediction of the allosteric sensitivity in the saturation mutagenesis experiment. Um, right? Which I might add, you know, there's been multiple saturation mutagenesis experiments done on PDZ3, and they differ because you know of simple reasons like you know different experimental conditions, but also because some of the experiments isolate just the functional effect on the protein, whereas others have both the functional and possible folding effects in there. And so the you know uh, this level of prediction of allosteric sensitivity is within like the differences in agreement between different experimental data sets. So there is really something there in terms of the dynamical fluctuations encoding correlated motion that is predictive of allosteric sensitivity. Um, we can also use this to really look at the total entropy of the protein and then compare that with calorimetry experiments, right? So here, just to situate here, so what we looked at is here's PDZ, and there's PDZ in a truncated form, and we can ask what is the binding entropy of PDZ binding to this its substrate, which is this cryptopeptide, right? Now, we would expect that because our method is only calculating the conformational entropy of the protein, what we would really want to calculate is what's called the delta delta S, which is the difference in the binding free entropy between the wild type and this truncated version of PDZ, because that would subtract out any other sources of entropy that may differ between you know in the bound versus the unbound state. For example, maybe the solvent entropy is different. It turns out that in this case, the conformational entropy is the dominant source. So we don't actually even need to look at the delta delta S to get a good prediction of the change in entropy, right? So the conformational this entropy tree using the formula that the simple formula that I talked about here, right? Just subtracting the mutual information, which is the double counting, right, of the entropy, right, from the entropy itself, from the individual residue entropies, you should subtract out the mutual information. You can get you can accurately calculate the binding entropy in this case of the wild type and the mutant, and it compares very favorably. For example, and the people have developed using NMR what's called these entropy meters, or estimates of what conformational entropy is. So this method does better on the proteins we've looked at than the NMR protein meter, right? So for example, the NMR protein meter does not do super well on the binding entropy of the mutant. But I think the most important comparison is looking at, again, the delta delta S. What is the difference in the binding entropy between the wild type and the mutant, which should subtract out the sources that are really not due to the conformational entropy. And there, the NMR meter is still is not doing as well. We also looked at the signaling protein, which is ER alpha, that I kind of motivated this talk with, this signaling protein that that recognizes estrogen, right? So we also performed this analysis on this protein, and in this case, the absolute binding entropies are not as good as it was in PDZ3. They're still reasonable. But again, the delta S between a mutant and the wild type in terms of its binding to its ligand is very accurate. So this method can directly calculate the conformational entropy of the protein to compare with calorimetry experiments. So now going back to I think a question that was asked at the beginning: should we expect then this information that's hidden in the dynamics, in the ensemble of the protein, to actually be able to reconstruct phylogeny, right? As I showed before, the structure does not. And so if we look again at this family of steroid receptors, what's really interesting, I think, just from a protein function and evolutionary standpoint, is that you have some ancestor fold, right, that can do some kind of signaling. That's evolved to do some kind of signaling, and now it needs to diversify. And instead of having to diversify to really change its fold in some way to recognize different kind of ligands, and also to have different allosteric pathways and different even activation modes, like some of these homodimerize and others do not, right? So there's all sorts of different kind of functionality and different kind of computation that's happening, and it's all apparently hidden somehow in the ensemble, because structurally they all look the same, right? And and there's also something where if you just look at fluctuations themselves, so if you look at the root mean square fluctuations, right, and say, okay, what if I just directly calculate fluctuations along the chain of the protein? Can I then, just using fluctuations, the signature of the fluctuations themselves, could I get the phylogeny? And that's not so simple. So here, you get more dynamic range, but looks like you're doing even worse than using structure to predict the phylogeny. But if you construct these entropy trees, right, which are these intraprotein maps of entropy and shared entropy, right, that does seem to actually encode the phylogenetic history, at least in this protein family that we've looked at. So we haven't looked at more than this family, but you not only get the dynamic range, but it looks like looking at how the entropy is actually distributed in the protein is actually like saving the memory of the sequence diversification. And so what's happening here that's invisible to looking at the structure of the protein is that the entropic network is functionalized and diversified, and is the variable that is sufficient to recover that diversity, right? And so you can get then really different kinds of functionality without having to innovate on your basic folding template. Which to me is kind of interesting because it didn't have to necessarily be this way. Okay, so in the last couple minutes, I just want to kind of touch on, I don't have enough time, and this is still in its early days of okay, so we saw that there's a way that we can extract functionally useful information from the dynamics itself. So we can extract the coupling in a protein, and we can predict the entropy of a protein from the dynamics, but that still doesn't answer the question. But what even allows this long-range communication physically in the first place? And I think the answer really is in the same source that allowed us to do this tree analysis that the first half that I talked about, which is really in the discrete nature of the dihedrals. So what I mean by that is that we employed, and this is the work of Alan Katz in the lab. So this is the atomistic model of a protein. You have these all the atoms are there, right? And you have to look at all their detailed chemistry and the chemical interactions, and you have long-range things like long-range electrostatics, etc. Right? What Alan did was he constructed a simplified minimal model in which you just have springs that's holding together the fold of the protein. But what is unique about his model is that there are no chemical details at all. Everything is the same kind of steric sphere. But what he's able to titrate in his model is that what he does is that he changes the degree of nonlinearity of the dihedral potential. So, for example, you can have a flat potential where the dihedrals just can rotate freely. You can have a single well, right? A single minimum, or you can have something that's closer to what proteins actually have, which is a multi-well dihedral. And he just asked the question, if I now have these different levels of nonlinearity in the degrees of freedom, is this sufficient to enable long-range communication versus not, right? And the measure of nonlinearity that he was particularly interested in, given the previous results I showed on atomistic simulations of how we were able to extract these entropy maps, taking advantage of the very digital or discrete nature of dihedrals, is that he came up with a metric of discreteness, right? So, for example, if you have a single well potential, by definition, there's no discreteness. You're in one well, so you don't have two digital states, right? If you have more than one well, so you have two or more digital states, then the level of discreteness is really determined by the height of the barrier. So if you have a height of the minimum barrier, okay, so that is if you don't want any of your barriers to be low, so that you're like very easily just transitioning between this basin and this basin, then it's not very discrete. And so the discreteness is basically if you have multi-well potential, you look at the well with the lowest barrier, and it's just the tanh of that barrier normalized by KT. Just a way to like get a proxy for kind of an intuitive notion of like how digital or discrete the degree of freedom is. Okay, I'm going to skip this for the interest of time. What he's found was that he can modulate how connected the network is by having fewer springs to connect the network, right? He can have soft or hard springs, so spring constants are either essentially rigid constraints or springs that are allowed to move. What that does is just titrates the level of discreteness, the level of emergent discreteness of the individual dihedral angles. Okay, but that level of discreteness is a really good predictor of the correlation length of which is measured by the distance over significant mutual information is transmitted between any two dihedrals. So what this preliminarily is looking like is that not only does the digitalness or the discrete nature of proteins enable us to actually be able to calculate this entropy map, because without this digitalness, we would not be able to map to this effectively much lower dimensional, non-continuous but discrete state and make a separation separation of scales in terms of strong coupling within residues and just pairwise coupling between residues, um, uh, contingent on these individual, well-separated, discrete digital states within. That allowed us to actually sample the entropy sufficiently within the amount of simulation time that's available in molecular dynamics simulation, right? But that same property seems to be what allows proteins to communicate, right? And it's kind of reminiscent of the advantages of digital versus analog communication in macroscopic telecommunications, right? So that you have a much more robust framework for communicating information and also for error correction using a digital versus an analog framework. So that's a direction where we're pursuing with more analytic theory in the future. But I thought I would kind of end on that and just summarize kind of what are the right collective variables of protein ensembles, and that really gets at physically, right? What are proteins, right? And so the structural degrees of proteins are digital. The digital nature allows convergent sampling of thermodynamic coupling using atomistic simulations. But the digital communication also seems to enable allostery. And it seems that information flow, at least on the proteins we looked at, is well approximated as a tree. And this tree gives us a compact, concise information structure that predicts total entropy, but also allosteric sensitivity, as well as encodes the phylogenetic information of protein families. So now the question is like, should we then more systematically then characterize protein ensembles on top of protein structures? Next to the static, the geometric or static chemical structure of protein, should something like this, like an information structure, be put alongside it to complete the description or the definition of what a protein even is? Because without that latter one, in many cases, especially in the case of signaling proteins, it just looks like a bunch of identical static structures, right? And not just static structures between proteins that do different functions, but even the same protein, oftentimes when they're bound to ligands and not bound to ligands, the protein structure looks the same. Okay, I think that's all I have. So thank you all for listening, attending in the morning. Thanks for all the questions, and I'm open for more questions.
We have time for a few quick questions.
Yeah, thanks for a very nice talk. When you're computing the phylogeny, are you using the entropic measures to get a distance between the sequences? And if so, how are you doing that? What is the distance between phylogeny through those measures?
Yeah. So we looked at different. We looked at it different ways. So one is we just looked at the difference between the mutual information part. So we just look at the edges and look at what is the differences between the edges. But the data that I'm presenting is more straightforward. We just looked at the entropy of individual residues. And we calculated the distance between, like, if you have two proteins, each residue has its own. Now we can calculate like the entropy of each residue, and now between the two residues of the protein, there is a distance. Like in this protein, this residue has this much entropy, and that other protein has that much, right? And then that's summed over the whole protein.
I have one question. I mean, the tree approximation makes some assumptions about how strongly correlated the system is.
Yes.
In your experiments or numerical experiments, how many distinct states would you say the protein is?
Yes. Let me go back to the. So we actually, it's quantified here, right? So I think I went pretty fast through this part, but basically this is looking at a collection of proteins. So that's why there's lots of residues. So this is looking at the proteins that we looked at. And, you know, the most frequented bin, right, is that the most likely bin of how many residues is between 0 and 5. So most residues just have a few states. And that's because many residues are like in a core, so they might have one state, maybe just two. And of course, there's some proteins that are, have small side chains, and so they have fewer states, right? But if you look at the cumulative, right? If you look at the cumulative fraction of residues, right, you can see that if you go up to 20 states, right, just looking at the residues that have no more than 20 states, you can get 90% of all residues, right? So that means that there are still a significant number of residues that occupy many, many states. But we haven't found any residue that occupies more than 40 something states.
I think maybe it's a slightly different question in the sense that you can have, let's say, 10 residues in 2 possible states. And globally, you can have just 2 distinct states, because you can have one state where all of the residues are in maybe the left state, and other global state where all the residues are in the right state. So that would be just 2 conformations, but 2 possible states per residue. So in these scenarios, do you see, yeah, I guess, is there a way to measure how correlated these states are?
Yeah, so we measure that through the mutual information. So for example, let's say this residue has 5 states, right? And this residue has 4 states. These are the actual states. You can see that each state has like a collection of overlapping structures, because those are like the structures that all belong to the same state. Okay, and now we can just ask, like, throughout the entire simulation, how frequently how frequently in the simulation is residue 1 in state 1 and residue 2 in state 4, and that would be the value of this grid point, right? And so we build up this joint probability distribution, and then we can calculate the mutual information using this joint probability distribution. So so that takes care of all the combinatorial states between all the between all the discrete states of the residues.
And maybe just follow up on that, then do you get a sense for, just like you were saying, that for every residue it's between 1 and 5 or something like that? Do you get a sense for a given protein, how many distinct conformational states you typally tend to see in these simulations?
Yeah, so we haven't counted that, because so that's interesting. So basically, according to the tree model, right? What's nice, and also definitely a approximation from the tree model, is that oops, where did it go? Let me just bring up the tree model itself. Yeah, the tree model means that the tree model actually produces the full joint probability of any possible combination. And this is an astronomical number of possible states, and it gives you a probability. Of course, that probability, so the vast majority, right, of combinations in this joint probability distribution is, of course, not sampled in the molecular dynamics simulation. The tree imposes that structure, right? So so we haven't gone back to actually literally count how many states are, like, how many distinct global states there are in the whole protein. But it is like, it is it's limited by like 5 microseconds of molecular dynamics, whereas the tree, even though it's produced from that, because it has these strong assumptions, right, of pairwise correlations only, it produces the full probability distribution of any combination of like states. You can say, you ask, like, what is the probability that the protein will be in this weird configuration? And in fact, many of these things, the tree will predict a non-zero probability, in which we know that the actual probability must be 0, because a large, I don't know, a significant fraction of these states will have atoms that definitely will overlap each other. Will be like a sterically unallowed, like energy blowing up state, right? The tree doesn't matter; it'll predict some non-zero probability for that. So that's whereas we know that like for a lot of these, because of this, right? Because when you propagate these things down, it's not then taking care of like these accumulated steric interactions that may propagate. So so what I'm saying is that the tree is not a great model for generating like a new ensemble. Like if I were to just draw conformations from this tree, many of those conformations will be like energetically illegal. But the tree is a good model for generating the entropy map.
Got it. Yeah.
Hey, Milo, it's Rama.
Hey,
just
Rama.
wanted to, just wanted to ask, I mean, there's all these discussion that questions you've been hearing from Akshit and Gautam are about like computing kind of the global energy landscape of the protein, right? So the bottom line is, from your model, you ought to be able to construct a free energy landscape for the protein. And of course, you have to choose effective like collective variables to look at it, right? But
Yes.
Because there are experimental data that actually one can compare to. The question is, do you see in such a free energy landscape that the protein basically lives in two configurations, right? Right. Underneath which there is all these residue entropies and discrete states that all the side chains can take, but collectively, what does it all roll up to, right? In terms of the energy landscape the protein lives in.
Yeah. Okay, yeah, good question. So here, yeah, I think that's a good point. I think we should do that. For these proteins that we're looking at, because they are not proteins in which, for the ligand state, there is a global refolding or global configurational change. I would not expect only two distinct basins, but that can still happen even if it doesn't obviously seem like that from the outset, right? So I think the answer to that will only be resolved when we actually do the analysis.
Cool. Okay, so then in the interest of time, we will stop now. I just want to thank Milo for not just giving a talk online, but then giving so at close to midnight, right, Milo?
I'm a night owl, so yeah, this is
okay. So
early.
thanks a lot for a great talk. Let's just give a huge round of applause to Milo, and then we'll break for tea.